All right. Hello, everyone. I see it's now 1 o'clock, so we'll go ahead and get started with the Aquatic Invasive Species webinar series for today. I'm Megan Weber. I'm an aquatic invasive species extension educator here at the University of Minnesota. And we're excited to have Matthew Barbour joining us today from United States Geological Survey to talk about his work with zebra mussel control using low-dose copper. I have just a couple of housekeeping items for you, and then I'm going to turn it over to Matthew for his presentation. Everyone is muted right now, and it'll remain that way for the duration of the webinar. But if you do have questions that you'd like answered, you can enter them in. We're using a tool called Slido for our Q&A. You can get there by going to slido.com, so it's slido.com, and enter the event code COPPER. I'll also have a QR code that shows up during that Q&A portion for you. You can enter those at any point throughout the conversation, but we'll wait until the end to get to them. And we are recording today, so if there's anything that you're hoping to revisit and see later, you can watch for that. It'll go up onto our YouTube channel. Usually just takes a little bit of time for us to get the transcribing done. That's all that I have for the housekeeping items. So I am going to shift now and let Matthew introduce himself and his presentation. Thanks for joining us today.
Hi, I'm Matthew Barbour here with the U.S. Geological Survey at the Upper Midwest Environmental Science Center in La Crosse, Wisconsin. So just downstream for you folks joining us from the Twin Cities area. I'm here today to talk about some of the research that the USGS has done in cooperation with MASERC, primarily in Lake Minnetonka and elsewhere, investigating low-dose copper control for zebra mussel populations. So on behalf of the rest of the research team that I'm a part of, Matt Melmans, Todd Severson, Jeremy Wise, and Diane Waller, I'd like to share some of our findings with you. So we've performed 2 treatments now on Lake Minnetonka. Some of you may remember some earlier presentations if you've been coming to these for a while. The orange bays you see there are St. Albans. I have a nice little cursor here, I think. Yep. St. Albans Bay, we treated in 2019, and we used Robinson's Bay as a reference site, which remained untreated. And in 2022, we treated Maxwell Bay here in the kind of the pink color, and we used this end of North Arm Bay as our reference site for that. There's the button. So St. Albans Bay, we'll just stick chronologically here, is about a 66 hectare body of water, fairly well hydrologically isolated from the rest of the lake. It is connected through a little channel underneath a bridge here, and that's its only connection with the rest of the lake. So for this, we decided to try a novel epilimnion, or just trying to treat the upper surface of the lake. And hopefully using that thermal stratification to help us keep our concentrations on target. We treated initially to a target of 100 micrograms per liter as copper, and we used 60 micrograms per liter, then sustained for 10 days over the course of 5 applications. The product that we use here is EarthTech QZ. It is a commercially available and EPA registered pesticide for zebra mussel control. So the treatment itself achieved an average of 83 micrograms per liter over our treatment window, which occurred in late July and early August of 2019. You can see here that, sure enough, we put copper in. We can detect copper on the backside compared to our reference bay. And of course, the copper, through various uptake means, dissipates, gets taken up by plant matter, organic matter in the sediment, and so forth. We continued to monitor our treatment into the following years, and we did notice some kind of spikes even in our reference site, indicating that somebody else is using copper. Copper is a very popular control tool, not only for zebra mussels, but also a lot of algae and herbicidal applications. So as far as our targets here, it's very, very important to note that this was not an eradication attempt. This was a population control attempt. Our whole scope or objectives were to try to reduce a population through reducing recruitment. So zebra mussels are broadcast spawners, and they have this larval stage called a veliger that's freely planktonic swimmer. And it settles out on the substrate, attaches, and then you end up with the mussels that we're used to seeing. Our idea was that if we can target these veligers and reduce their numbers, we reduce the number of mussels settled and therefore recruited into the next generation, ultimately ending up with some kind of population control approach. So as far as these veligers go, number of veligers per liter, you can see our treatment did greatly reduce the number, or appears to have greatly reduced the number, not only in the treatment year, but also in the 2 following years. However, when we come over to actual settlement on plates, we saw very little settlement the year of treatment, a little bit of settlement the next year, and then it kind of slowly ramped up to be comparable to our reference site. So the good thing is, I mean, we nailed the timing, we think, because of this peak in veligers in our other bay. And then, we observed this peak indicating that we had hit the right time of year for this. And of course, there's always going to be some that slip through the cracks, which is the big problem with eradication attempts. And then the kind of added bonus is that we saw reduced settlement even into the following year, which we were not really expecting. And on top of that, with our seeded mussels, we saw around a 30% adult mortality rate in the zebra mussel population, which was much higher than we had anticipated. So kind of an added bonus, but we'll get into that a little later. All treatments come with trade-offs. The biggest trade-off here was our zooplankton community. The zooplankton community did take a bit of a hit, not only in overall abundance or number of zooplanktons per liter, but also diversity decreased too, with Daphnia being kind of the hardest hit out of the whole group. But they do bounce back pretty quick. We think because of their life cycles and reproductive cycles that they're able just to repopulate faster compared to zebra mussels, which breed, typically once a year in these kinds of lakes. Zooplanktons are, reproducing continuously, more or less. So the key takeaways from St. Albans Bay is we did indeed see veliger settlement and suppression even for that additional year, which was great. We did see some impacts to native communities, particularly those zooplanktons like I talked about. We did also observe or monitor the benthic invertebrate community, but those were really stable. It didn't appear like there was any huge shifts or anything in the community or anything alarming when we compare it back to our reference site. And then, of course, the nice additional bonus that we had higher than anticipated adult zebra mussel mortality. So if you recall from a couple slides ago, we were treating at like 83 micrograms per liter was our average through our treatment period. Some previous work we had done with MESERC in our environmental chambers, trying to figure out temperature and duration of these various compounds. EarthTech QZ dose response curve here indicates that we need about 4 milligrams per liter, which is quite a bit more than the 80 micrograms per liter. We're talking orders of like 2 orders of magnitude almost. So that's quite a difference. So that's how low we're going, and we're seeing these kinds of results. It's important to note that this is 2-week exposure, where ours was 10 days. So direct comparisons are a little shaky on that front, but still the numbers are drastically different. And we were very pleased to see that we had killed that number of zebra mussels. So then moving on to our next treatment, Maxwell Bay, the idea behind this was that we would attempt to replicate the success that we saw in St. Albans on a slightly larger scale, so 125 hectares instead of 66. We also modified our application technique to only hit the shoreline. So we made one pass around the lake. We launched from down here in the south end. We made one pass around the lake for our applications, following the shore, approximately 100 meters, 150 meters or so out from shore, and made our applications. Here the target was 50 micrograms per liter as copper over a 12-day period with 4 applications. So here again, our copper concentrations, 50 being our target. We kind of fell right in there. Again, we were targeting that same time frame. So late July, early August, right when we anticipated seeing the most veligers is when we wanted to get our copper in the water, so we had a chance for the highest effect. Just like St. Albans, highly variable copper concentrations during the treatment. And then again, several spikes and high points, even in our reference sites for copper, likely being used again for herbicidal or algacidal treatments. Veligers, here we were measuring the year before. We slightly altered our sampling regimen to be more frequent. So we were collecting a lot more samples throughout the year to try to catch some of these smaller peaks and shifts in various communities and so forth around our treatments. But the year before we treated, you can see both bays, very similar densities during those peaks. Timing was a little shifted. That might just have to do with sometimes even the wind direction can alter some of this stuff, but the bays are slightly different. So it's not too terrible to see a slight difference like that. Into our treatment year, North Arm, our reference site, you can see that beautiful spike there indicating, yes, indeed, late July timeframe is when they were really abundant in the water column. And that indicates the perfect time to try one of these veliger targeted treatments. In Maxwell Bay, right around the time of our treatment, you can see those veliger numbers dive right off, which is perfect. That's what we're going for. And then the following year, again, those numbers stayed kind of low. So very similar trends to what we saw in St. Albans Bay. We don't see quite the nice big spike in North Arm, but again, we saw those other copper treatments occurring, or we detected other copper sources, at least, that may be influencing that. So very similar to St. Albans in regard to veliger trends in our treatment area, but when we look at settlement, it was a little different. So the number of mussels per square meter, very comparable in that pre-treatment year, extremely low, about 90% reduction or so in zebra mussel settlement the year of our treatment. And then we see it bounce right back. So not exactly sure what that is. Maybe we missed the peak in our peak veliger production in our veliger sampling the year after a treatment, and we just didn't capture that nice peak. But either way, the whole idea was to reduce settlement. We did it the year of. We just didn't see those favorable results the following year like we did in St. Albans. We also monitored biomass of the mussels settled on the plates. Pretty comparable the year before. Staggering difference, right? Of course, if you have less mussels on a plate, you're going to have less biomass on the plate. This is an indicator of that. So we were able to reduce zebra mussel biomass and then again, it picks right up. So those two trends are very, very tied together. Zooplankters, so our more frequently sampled zooplankton community, we did pick up some more of those little nuances. So as you can see in our pre-treatment year, it is pretty, I guess the right way to describe it would be chaotic. But it's very dynamic, always changing, the zooplankton community. I would hardly call it stable. But we can see it's kind of clustered here around diversity index of 1. So we'll call it stable for all intents and purposes today. Into our treatment year, we see again it's pretty comparable between our two bays. And then again, late July, early August, we treat, and those numbers decrease. So we see a decrease in zooplankton diversity indices following a copper treatment. No big shock. We know that copper is a well-known intoxicant to much of aquatic life, and zooplankton are one of the more sensitive species out there, which is why we monitor them during these treatments. The one thing here is that with our more frequent sampling regimen, we were able to capture a really nice recovery by September, indicating, these zooplankton communities might take a bit of a hit on the nose immediately following a treatment, but within 2 months or so, they're back to where they were, more or less. And then the following year looks a lot like our pre-treatment year. Both bays kind of hovering around the same indices. So key takeaways from Maxwell Bay: veliger suppression was achieved, but not quite like we saw in St. Albans. Settlement bounced back the next year. It might have to do with more hydrologically connected Maxwell Bay versus St. Albans, meaning that there's just a higher influx of, or higher potential for influx of veligers from outside of the bay to come in, and stuff like that. There's just a lot, when you step out of the laboratory into the field, there's just a lot of variables that are really, really hard to account for. But the big thing is we were able to achieve that veliger suppression. It just wasn't quite like St. Albans Bay. Again, zooplankton rebounded. This time we had the more frequent sampling data to kind of back up our assumptions on that. And then one thing we did notice is that the shoreline treatment, while we were only targeting the shoreline, we had essentially treated the entire bay. We were detecting copper throughout the entire bay shortly after our treatments. Now, the concentrations around or in our path, of course, were higher right after treatment, and then that dissipated. But with just simple diffusion and wind and wave motion and so forth, it's going to mix pretty well throughout the whole thing. So just something to keep in mind when you're thinking about pesticide application and drift. In the water, it's almost guaranteed to drift unless you have some kind of barrier in place. So it's just something that managers should maybe kind of consider when they're looking at treatments like this. So if you recall back to St. Albans, we had that higher than anticipated adult zebra mussel mortality event. And that really kind of got us thinking, why? And if it's something that we can account for, maybe applications during this time frame can almost be a kind of a two birds for one stone transaction, where you're targeting veligers and reducing their numbers, thereby reducing settlement, and you're also hitting the adults at a time where they're most susceptible. So you can kind of double-edged sword hack through that population. But we wanted to see what was going on. And our original thoughts were that it's tied to either temperature, which is regulating their metabolism, or reproduction. So there's a lot of stresses with reproduction in our selective species like zebra mussels, where they're putting a lot of resources into reproducing every year. The literature states that up to 1 million eggs per female in a given year. So they're really putting a lot into that reproduction every year, and that can be very taxing on the organism. So we went to Robinson's Bay in Lake Minnetonka, collected mussels from there, and we held them and exposed them at ambient lake temperatures back in the laboratory. We also investigated and tracked their reproduction status and condition, so their tissue mass but to shell length ratio and their total lipids and stuff like that as well, just to try to get an idea of their condition. We expose them to copper in 3-liter vessels in temperature acclimated water with aeration. The exposures were all 96-hour static exposures, followed by 96-hour recovery and flow through conditions. And that's just so that they get a chance not only to die, but also recover from any potential narcotizing effects and anything like that. Pretty standard tox trial. We did 7 of these exposures throughout the course of the year. And we really lucked out this year on weather because we were able to go up on, I think it was Mondays we went up and collected. And then we were able to initiate the exposure exactly so many days later and everything and stay right on track. So the pre-holding conditions and everything were the same across the board, which was amazing. Made it very easy to justify. So as you can see, of course, this is a bioassay, so we're selecting a certain size class of zebra mussels. Shell length shown here in purple stays stable throughout because that's the size class that we're selecting. But for that particular size class, we see a decrease as the year goes on in tissue dry mass. This is straight up because they're releasing gametes. Their metabolism is also higher. They're burning through energy stores, but this is also when water's very productive. So they should be eating more and everything like that. Peak filtration rate and so forth is also right in the same time frame. So we really are starting to see a case for reproductive stresses emerging simply by looking at condition. On the right is just a simple contrast between those two parameters shown on the left. And we did detect that there were 3 distinct groups. Two distinct groups. The early season, and then sure enough, once you started hitting around mid to late July, all of the slopes changed. So that's right when they're reproducing. So everything is kind of lining up with reproduction being the thing here. Same thing with the dose response. So we have copper concentration along the x-axis, mortality as a proportion on the y-axis, and then our dates of all of our exposures are color-coded here. And we see this shift. So very early in the year, we didn't even achieve 100% mortality in our tests. We were only able to model up to maybe an LC50, and even that was extrapolated a little bit. But as the year progresses, we see it march over here to the left, which means zebra mussels require less and less copper to achieve the same mortality rate, which is a good thing. That means less chemical is needed to kill the same amount of zebra mussels, which means less chemical in the water for non-target impacts, less chemical required for purchasing, so decreased cost, things like that. Kind of unfortunately, we only kind of capture the beginning of it, but it starts to creep back to the right, kind of right around that September timeframe. So post-spawn, kind of when we're wrapping up our field operations and stuff like that. So I emphasized the reproductive status of these mussels a lot, but For those of you familiar with zebra mussels, we know that their reproduction is highly correlated with ambient water temperatures. So trying to tease those two apart is very difficult to do. We attempted to use some serotonin to spawn out the zebra mussels to induce a spawning event in our lab. We either had collected them just a little too early to mature their gametes all the way through release, or we just didn't hold them long enough in our serotonin bath. We were worried about overdosing them, which then can end up confounding our results. But basically, we were unsuccessful in teasing apart reproduction versus temperature in that regard. But knowing that they're correlated, we can see temperature on the dotted line here. And you can even see the same little trends in our LC90, which is the concentration required to kill 90% of zebra mussels. You can see like the same little trends emerging in both. So yeah, we still aren't exactly sure what the cause is, but we know that they are most susceptible in late July, early August. So that lines up with the greater than anticipated mortality event that we saw in St. Albans Bay, and as an added benefit for anybody attempting to do a zebra mussel control effort. This really spells it out, particularly when we look at events outside of the area, say like Highline Reservoir in Colorado. They attempted to do a treatment very early in the year, and they were very, very unsuccessful. They had to end up drawing down the entire reservoir and letting it go dry, which out west is a big deal. But here it just shows if you want to hit them with copper, the story's really coming together on when to do it and how much to put in there. So putting together everything, which I kind of did a little preemptively last slide, copper at low doses appears to be effective, particularly at targeting veligers. And we're starting to kind of refine some of the nuances with application as far as doses and timing. This is starting to shape into a potential control option, unlike the traditional eradication or bust mentality that's out there. This really has a different place where it kind of fits in. So for whatever reason, zebra mussels never really seem to have settled into an integrated pest management system. And maybe I'm way off on that, but there really seems to be this entrenched mindset of what do you want to do with zebra mussels? Well, we want them gone. But eradication really is the exception. It's not a realistic goal for any of this. Like any other pest, we go out with a very objective mindset, something very specific we're trying to do. We're very good in the integrated pest management scheme already. We're very good at identifying the pest, Tricinid mussels, right? We know that they're bad. We have a lot of effort into preventing the spread, which is great. We should really be keeping that up. We can monitor and assess populations pretty well, but what we don't really have yet is this threshold for action. When it comes to early detection, rapid response, we seem to have that ironed out pretty well. But for an established population, it seems like once they're established, nobody wants to touch it. So that's kind of where our research is trying to get into, is like, is there something we can do to help restore ecological function in these water bodies, which brings in this whole concept of functional eradication, which has been used in other invasive species, such as the green crab and what was the other one? Lionfish. And I think there's a type of crayfish out there that they've tried this with. But the whole idea is that instead of eradicating them, you just dial back their population. You kill enough of them to where their population can't have the same ecological impact, or they're not functioning in the ecosystem the same way through their population, which I think would be a good fit for zebra mussels because we know it's a density-driven effect. So on the right side there by the green check mark, we really know what insufficient suppression looks like. There's rapid loss of ecological integrity in systems and so forth like that. We see plant growth, harmful algal blooms. I mean, all of that is really well documented. What we don't know, and this kind of feeds into that threshold for action too, is what happens when we dial back that invader density. Is there a point where that zebra mussel population no longer has that same ability to affect ecosystem function? So that's kind of what we're after, trying to investigate some of that and figure that out. Because if straight up eradication isn't an option, population reduction is the same as any other pest. In agriculture, agriculture has a lot of great examples for pest management. The farmer doesn't go out and apply pesticides to his fields thinking he's going to eradicate the weevil. Farmers are going out there and doing it at a very specific time with a very specific product with a very specific objective, something that's measurable and achievable, the whole gambit. We're just trying to figure out what some of that might look like still. And that takes us into our current state of affairs with these projects. Structured decision making, which has been used pretty successfully, it seems, in the realm of invasive carps. We're trying to roll it in and apply it to dracinid mussels in the state of Minnesota. So we've partnered with MACERC on this, Minnesota DNR, multiple other stakeholders, managers and researchers, all trying to get together and figure out what exactly matters the most in zebra mussel management, and kind of like how can we get there and what does it look like, and just really trying to take focus. It really is it can be a very complex thing. It took us 2 days sitting in that room to kind of hammer out the beginnings of a decision-making model, which, by the way, doesn't just spit out an answer. It spits out a couple of options that fit the values that you fed into it to begin with. So it's a dynamic thing. It's not a one and done kind of tool, but it's getting there. It's starting to take shape. So out of this SDM, or structured decision-making process, we identified another potential lake for treatment, Lake Riley, and that's in the, I got it written down here. The Riley Purgatory Bluff Creek Watershed District. Sorry. And they've been very helpful in kind of helping shape some of this as well, and giving us feedback and the community outreach that is necessary to pull off projects like this. But we're still kind of in the baseline monitoring phase. We have samplers out right now. So if you frequent Lake Riley and you see us out there, give us a wave. But yeah, right now we're just working with stakeholders and trying to see what this is going to shape into. But the whole idea is to try another population suppression. And what's really nice is Lake Riley has a lot of pre-existing data from previous years that we'll be able to incorporate. So we have a larger snapshot of all these various parameters over time. Hopefully that'll help us to really identify which parameters are coming back like they were pre-invasion and stuff like that is the goal. So I think I'm a bit early, maybe right on time, but these projects represent a huge undertaking. I just want to say that, and there's been tons of people working behind the scenes. I'm the lucky one that gets to sit here and bore you all with my voice, but the whole team here at the USGS, the whole team at MACERC, our partners through the Invasive Mussel Collaborative, all the various state, local agencies, counties, conservation districts, watershed districts, even local businesses, marinas, and so forth. And of course, funding from and the Environmental Natural Resource Trust Fund, and GLRI, and just tons of people highly interested. And the numbers in this, the number of attendance in this right now, it just shows like there's a lot of people interested in this, and I think it's a really worthwhile endeavor. So hopefully this partnership between USGS and MACERC continues on to produce some great things. I think there's a lot of work to be done. So with that, I will welcome any questions, I guess.
That's great. Thank you so much for all that great information. I'm going to throw up a slide here for those of you with some questions. I see some have already started to come on in, but if you'd like to join the Q&A, you can either scan that QR code with a phone or a tablet, and it'll bring you right there. Otherwise, you can go to the website at slido.com and enter the event code COPPER. And you'll be able to view the questions there. You can also upvote existing questions in the queue. So if you see one that you are particularly interested in and want to be sure that it gets answered during the time we have for Q&A, you can upvote those questions as well. I'll leave this slide up for a little bit and then I'll pull it down so you don't have to stare at just the QR code and you can maybe see the people who are talking to you. But we'll go ahead and get started. There's one question that is already starting to get upvoted here. Wondering if there's a risk of zebra mussels developing a resistance to copper.
That's a good question. I know there are some examples of like transgenerational plasticity to copper resilience in other gastropods, or in gastropods, other mollusks, I should say. But I don't think there's been an example of it in zebra mussels. That's not to say that it's impossible. Certainly, anytime you have a pesticide, you run the risk of that, I think. So that's a really good question, and something that is a bit difficult because there's so few treatments that have been done. So.
Great. Always more to study.
Yeah, always. Every time you get an answer, there's 10 more questions.
Yes, I think we've got more than 10 for you here. Okay, next one was wondering if there's studies that have been done about how copper treatments might affect native mussels.
Yes, we actually had alongside of our native or our invasive mussels or zebra mussels in St. Albans Bay, we had native mussels. And I think we saw, and if Diane is in here, maybe she can chime in on this if she remembers, but I think we only had one or two that died out of all of them, and they were from the reference bay. But the other factor to really think about with it too is like copper. When you put it in the water, it's not going anywhere. And mussels, out of a lot of different things, mussels tend to really suck up in a lot of heavy metals. So we do see tissue concentrations of copper elevated in those species. So we were using fat muckets, I think they were, and yeah, they did increase their tissue loads of copper, but we didn't see the mortality.
All right. Next one to work its way to the top is wondering if this product or a mix like it could be used to decontaminate things like wake boat ballast tanks.
I think they may already do that, I think. But yeah, I mean, you'd have to adjust the dosage because of your contact times. And there is another product out there, another copper-based product, Natrix, which is kind of gaining some popularity after the treatments out in the Snake River. We've started some research with it, particularly with veligers. But yeah, it's just kind of we don't have enough data to really tell at this point. But I think it could be a good ballast treatment option.
Great. And I'm going to jump back to the native mussel question because Diane is present and just sent a chat in. Diane said that the native mussel mortality was insignificant. She says
Yeah,
there was slowed growth in juveniles right after the treatment, but the growth caught back up after 30 days.
yeah, yeah. See, I knew she'd remember.
All right, great. Next question was wondering about if there could be some coordinated effort. So, for example, if using a product like this to control algae, are there opportunities to coordinate algae control with zebra mussel control?
I think the possibility I think is definitely there. Um, a lot of it though might come down to the formulation of the various products. So like Earth Tech, it's labeled for dracinid mussel control. Um, but then you take an herbicidal product, even if it's made by the same company, and it's not labeled for dracinid mussel control, you can't apply it with that being your objective because of current restrictions and FIFRA and Environmental Protection Agency and all that fun law stuff. Um, but kind of like Bob Ross, happy little accidents. Um, I don't think anybody's going to lose sleep if a bunch of zebra mussels just up and die overnight, which is kind of a nice thing about doing this kind of research. Um, we don't really tend to make people upset by killing zebra mussels. So um, I think there's a possibility there. A lot of it comes down to the formulation and what it's labeled for, though.
Great, great. Okay. Um, Next question is wondering about what other species might be harmed by copper treatment. So I think you talked about some of these, and there might be a few a few questions kind of along this same path. So just in terms of other species, there's questions about fish and plants. We've talked about native mussels a little bit. I saw Eurasian water milfoil as well in further down the question
Mm-hmm.
chain.
The biggest thing with the sensitivity of these other species is the lower concentration that we're using. Um, Copper is a very well studied contaminant in aquatic biology, so there's tons of information out there, especially with the EPA's uh, registration eligibility decision, the RED document from I think 2016 or something. But uh, there's tons of toxicity data out there. The thing is that we're using 50 micrograms per liter, where a lot of those other things are permitted up to 1 milligram per liter. Um, so we're treating, what's that, like 10%? Call it 20% or whatever of the labeled maximum dose. Very small compared to what we're allowed to do. So I think that really kind of negates a lot of our non-target impacts as well. It's just given the size of the organisms and their habits, zooplankton seem to be the most sensitive to it. So I hope that kind of answered the question.
Great, thanks. Yeah, and if there's any more maybe specific parts that someone's interested in,
Yeah.
I'm sure they can get added into the question chain here.
Yeah, I don't have
Um,
a lot of non-target information like up in front of me. I know it's out there. Yeah.
sure. Next, we're learn about if there's longer term monitoring results to looking at beyond the 1 or 2 years post treatment.
What we have on the screen is what we have. It'd be nice, of course, to have longer term monitoring or even have monitoring over maybe multiple treatments over several years to see what long term management might look like. But it just really comes down to funding, basically, funding and time. It takes a lot to put these crews out on the water and process these samples and stuff. So um, we try to get what we can get and address as many questions as we can along the way. But at the end of the day, we can only do so much.
All right. Next one was wondering, what's the benefit of using copper versus some of the other control methods that might be out there? Potash was the example used in this question.
Right. So one of the first and most successful zebra mussel treatments was Millbrook Quarry, and they did use potash. That takes and still does a special permit, application use permit from the EPA directly because it's not a labeled pesticide product. Um, so the reasons behind using copper isn't necessarily because copper is best. It just happens to be maybe one of the better tools that's labeled right now. Um, so it's easier to get permits using something that's already permitted for use. But hopefully we can be getting some other uh, control tools online in the near future. Because ideally we're not just treating everything with copper, right? That part of that integrated pest management is multi-pronged approaches for control and stuff like that. So we're working with a pretty shallow toolbox, but hopefully we'll get there.
All right. Um, this question's wondering about how you selected which lakes became part of this project.
It's a good question. A lot of it comes down to uh, local support for it. So around Minnetonka, a lot of these bays have their own associations and so forth. Um, and then there's also, what the DNR ultimately will allow us to do. We might be a federal agency, but it is the state's waters. So we play by the rules, and that's just how it goes. So ideally, we'd be selecting different lakes, but we have what we have, and it's better than nothing.
Okay. There's a question then next wondering about if there's about phytoplankton. I think you might have touched on this one too, but about data on phytoplankton response to the treatments.
Oh yeah, I think I might have skipped a note on that because I wanted to mention in the St. Albans Bay treatment, we did notice an increase shortly after treatment in chlorophyll A, which was our only measure at that time on phytoplankton communities. Um, and I was at a NALMS conference, I think up there in the cities, and I was talking with somebody who was in charge of managing Western reservoirs, and they said they've seen similar trends, although they weren't sure of which species was kind of moving in. Um, Unfortunately, we didn't capture the right kind of data to answer the questions, but we do have some phytoplankton community data from Maxwell. It just hasn't been through the ringer yet, so to speak. Um, Stats, that is.
Great. So maybe some more information to come there.
Yeah, yeah.
Sounds good. Okay, I'm gonna stop the screen share so that it's not a giant QR code in front of everyone's screens, and we'll put the information into chat just in case you don't have it yet. If you have questions or want to keep up voting, just give me a moment while that rearranges my screen as well. All right. So moving on to the next question. Um, Looking forward into the future, do you envision some type of common zebra mussel treatment option within, say, like a 5 to 10 year time range?
That's really hard to say. It comes down to the continuation of this work and ultimately what objectives managers and stakeholders are after. I don't think there's ever going to be the one size fits all option. There just rarely ever seems to be something like that. The whole silver bullet idea. Um, but I think the more research we put into this, the more options will come out. I think. Um, right now, we know that hey, if you want to go out and reduce your settlement in a given year, I feel pretty confident we can repeat this, and hopefully we can do it again in Riley Lake Riley and so forth. And we're not the only ones doing copper applications too. There's been David Hammond from Earth Science Laboratories has done quite a few of them with a lot of success too. So I think there's growing information to help in better inform decisions when it comes to that. And a lot of it really does. It's going to come down to what your objectives are. What is that measurable outcome? Is it um, something to do with water clarity that might change your application technique or timing, um, stuff like that. So there's a lot of parameters outside of the laboratory, that's for sure.
Yes, and we'd all love that crystal ball, right?
Right? Yeah, yeah. I think I'd
Um,
even take a magic 8 ball.
All right, we have another question that's kind of moved its way up to the top, wondering about what the potential is for copper to accumulate in lake sediments over the long term and if there's any potential impacts from that.
It's a great question. It is a concern, but when we look at other lakes, because copper's been used so long, so Lake Mendota over here in Wisconsin, down in the Hara chain in Madison area, they still have large amounts of copper detectable in their sediments from pests or from herbicidal treatments with copper that took place back in the early 1900s. The thing about copper is once it binds to a ligand, it's very hard to cleave off of there. Oftentimes, so like in our samples, we want to cleave all the copper into free ions, right? We have to acidify it below a pH of 2. So if that were to happen in your lake, I think copper is probably going to be the last of your worries. But there are possibilities for it to remobilize in the food web and so forth. I'm not too well versed on that, but again, lower doses trying to mitigate some of those concerns as well.
Sure. Great. Thank you. All right. Next question is wondering if you're aware of any zebra mussel lifestyle differences, or sorry, life cycle differences between shallow versus deep water lakes.
Um,
Maybe to add on the example that they gave, wondering if, for example, your mussels are more pervasive in one versus the other.
oh, that a lot of that comes down to just how productive a system is. It seems like some water bodies go into and you get that big boom or bust, traditional kind of invasive species boom and bust kind of cycle. Other ones you'll see them like slowly creep up. Um, I do know that there's life history differences between like monomictic and dimictic lakes, things of that nature. So you go down to lakes in like Texas, they can reproduce multiple times in a given year, whereas you come up here to these northern stratified lakes, dimictic lakes, you get this nice peak like we're able to get. So that's going to influence your treatment strat or potential treatment strategies a lot, um, as far as what you're facing, and it really boils down to like know your pest, right? You have to know these things about the population you're going after in order to be effective. Um, Another perfect example would be uh, the Colorado River. They're seeing quagga mussel veligers year round. So there's definitely differences out there, and it's good to pay attention. And that's part of that monitor and assess step in IPM for sure.
Yeah, great. All right. So with the work that you've been able to do, have you been able to determine what any kind of like tripping point for when to treat for veligers might be?
It would definitely be that in a lake like this or like we have done. I would definitely say that late July to early August. That's when they're in the water. That's a good time to hit them.
All right.
Yep.
Okay. And then there's another question wondering if the dissolved organic carbons or hardness was measured during the studies, noting that a potential influence on copper effectiveness depending on those parameters.
Right. So that's getting at the biotic ligand model. Um, so increases in dissolved organic carbon is going to decrease your copper effectiveness. Um, there's a lot of information out there and some really cool tools that we've tried to employ. Um, as it stands right now, there is no biotic ligand model specific to dracenids. Um, I've talked with, I forget his name at the EPA, but he's in charge of kind of the BLM is kind of his baby to babysit and update, and he was interested in kind of updating that. So if there's any grad students in the office or in the lobby here, possibility. Um, but yeah, DOC is a big one. pH is a big one. We did monitor our other dissolved major ion constituents, so magnesium, calcium, sodium, and potassium as well, because those will compete for binding sites. Um, the thing is, though, working in these lakes, a lot of those parameters don't really shift around. It's pretty steady state. So comparing between water bodies is probably more important to know those things. We did capture the data so that later on, hopefully we can do that. But yeah, very good question. Important stuff.
Great. Okay. And then there's a question going back to the effects of temperature on there. So I think the
Mhm.
question is wondering if it's like the mussel feeding rate that's affecting um, how effective copper is on the mussels, or like why you might see those shifts in effectiveness depending on the temperature
I
or
mean,
time of year.
they are poikilothermic, so they're completely at the mercy of their ambient surroundings when it comes to temperature regulation and so forth. So um, while feeding is directly influenced, it's more metabolism driven. Um, yeah, that's really it. I mean, their feeding rates are up. So like when it comes to other delivery methods like an oral route, you definitely want to know that kind of stuff for sure. Um, but again, it's really hard to tease apart with something that's so dependent on temperature swings. It's really hard to tease that out of it, kind of like with the reproductive status. Really hard to tease apart. Like, would we ever be able to get a zebra mussel filtering at a high capacity at a cold temperature? Might be able to do it in the laboratory, but we'll probably never see it in the field.
Sure.
So,
Right. All right. And then it looks like we have some folks in attendance who are members of lake associations or other community groups, and they're wondering if there's any handouts or summaries or anything that they might be able to use to share in newsletters or other ways to reach others on their lake with some of this information.
awesome. Um, we do have two papers in the works right now. They've been accepted. They're in press, like just waiting to see the proofs, and then they're out the door. Um, coming out in environmental management. One is for the zebra mussel population in our target species with the treatment in St. Albans Bay, and the other one is for the non-target. So they're kind of a pair. Um, as far as a handout, I don't think we have anything specific to copper worked up. I know there was a handout that covered, I think it was all invasive mussel stuff. Diane was one of the co-authors on it. It was an informational handout. She might remember.
Right. Well, yeah, and I'll also add, we're recording. So in a week or two, you'll get an email with a link to the recording of this webinar. And if anyone wants to share that, they are more than welcome to do that as well. I see we're down to just 1 minute. Oh, and I see Diane made a note that we can follow up too. So we have the list of emails. So if a paper happens to come out and the proof goes out soon, we can be sure to follow up with that. Or if you have
Yeah. Well,
links to those, we can send that in the follow up to this as well. Yeah, go ahead.
And MASERC also has project pages, so does USGS. If you jump around between those two locations too, you'll find some more information as well.
Great. Yeah, I'm sure we can find those too and send them in the follow-up. I'll go ahead and do one final screen share on my end. Thanks so much for joining us. This has been really informational, really great to learn some updates about how this project has been doing and where we're at. As I mentioned, we've been recording this so for those of you online, if you want to revisit something or want to share with someone else, you can find that on our YouTube channel. To get there, you can go to z.umn.edu/AISTube. And it does take just a little bit for that to go up. We transcribe all of our videos, so give us a week or two, and you'll also get an email when that's live. Or you can feel free to subscribe to the channel, and you'll probably get a notification from YouTube when we post something up as well. Um, there is a survey as well. You should get a link when you close out of Zoom today, and we'd really appreciate it if you are able to take some time to complete the survey for us. It helps us with planning these webinars and improving any place that we can. I also have a couple of contact options up here. If you have more questions for Matthew, you can see the email on the screen there. If you have questions for us about the webinar series or our programs in general, you can email us at aisdetectors@umn.edu. So if you need some of those follow-up links from the webinar or anything like that, feel free to reach out to us there as well. That brought us to 2:01. So I'll go ahead and say our final thank you. So thanks to all of you for joining, and thanks again to you, Matthew, for speaking with all of us today. We really appreciate all of you joining, and we'll hope to see you next month. We'll have our next webinar about mercury concentrations in fish species in zebra mussel infested waters. Thanks, everyone.